Hi, everyone. This is Jonathan Cedillo, student at Grand Canyon University, and today I'm presenting to you life threatening diarrhea by Clostridioides difficile infection. Presentation for the class Biology 525, Concepts of Medical Microbiology, Professor Sandra Barrera. Background: Clostridioides difficile, bacteria that can confer life threatening diarrhea. One of the main reasons that this disease is contracted is due to the uncontrolled use of antibiotics. Usually, the disease presents in elderly people who are 65 years of age or older that are under antibiotic treatment. People who spend long periods of time in hospitals where the pathogen can be more latent can be also prone to contract the bacteria. And in people that have immune depression or already contracted the bacteria in the past. Symptoms of the infection include diarrhea, loose watery feces for several days, fever, stomach tenderness, loss of appetite, and nausea. Additionally, C. difficile represents a large health danger. Statistics show that during 2017, there were 223,900 individuals that presented the infection, in which 12,800 lost their life in the United States. Furthermore, in 1975, John Bartlett's research was concentrated in the issue of antibiotic related diarrhea and pseudomembranous colitis. This research led the scientists to the discovery of Clostridium difficile, hence the identification of symptoms, causes, and hospital related cases. Taxonomy: the taxonomy of the prokaryote Clostridioides difficile is as follows: kingdom, bacteria; subkingdom, Posibacteria; phylum, Firmicutes; class, Clostridia; order, Clostridiale; family, Clostridiaceae; genus, Clostridium; and species, Clostridium. Here, we can see a structure of the bacterium C. difficile. We can clearly appreciate that the bacterium has a rod shape. Also, the bacteria has a flagella, and they usually use this for locomotion. On the right side of the screen, we can see a piece of an infected intestine from a patient. The looking like type blisters, yellow, are colonies of the bacteria that has infested the intestine. How does it spread? The pathogen of interest can invade the human body through bacterial spores located in feces. Outer layers of objects can be contaminated with the spores of the bacterium due to the poor hygiene of people. As already explained, antibiotics are a causative agent in the development of C. difficile. In addition, the use of proton pump inhibitors play a critical role in its multiplication, hospitalization, immune deficiency, and elder people are also prone factors to acquire the infection. The identification and diagnosis of the pathogen is through cultivation, DNA testing, or antigen testing. When patients present invasion of C. difficile, but they are asymptomatic, the phenomenon is known as colonization and not infection. On the right side of the screen, we also can appreciate an infected length of the intestine. Treatment and prevention. To treat Clostridioides difficile infection, or CDI, A patient can ingest vancomycin, which can alleviate severe infection. Also, surgical review is advised in individuals presenting fulminant colitis or those which case is growing after treatment. However, therapies against chronic CDI is still a challenge for scientists. Furthermore, in order to prevent infection of C. difficile, individuals can use antimicrobial and one that maximizes therapeutic effect of the antimicrobial agent while minimizing the development of antibacterial resistance. Infection repression techniques are also used, decontamination, and personal care, including hygiene. Isolation, the use of personal protective equipment, thoroughly hand washing with soap, specifically if people work in hospitals or every time we visit hospitals. According to scholars, the use of soap and water to clean our hands has demonstrated more efficacy against C. difficile than antibacterial gel. Antibiotic resistance. When it comes to bacterial infection, a topic that represents a challenge for scientists is antibiotic resistance. This phenomenon occurs when pathogenic agents such as bacteria acquire protection or immunity against antibiotics, meaning that drugs such as penicillin won't work against infectious bacteria that can cause diseases, which are a problem for the human health. Bacteria, such as Clostridium difficile, has already presented this challenge, and some antibiotics are not an option to eliminate the invasion from the organism of target. Furthermore, antibiotic resistance can be developed in different ways, but in all instances, bacteria develop methods of defense against agents that compromise their integrity. According to studies made in feces, samples of different organisms, including birds or poultry, cattle, sheep, goat, camel, and buffalo, were examined to investigate the prevalence of Clostridium difficile across the samples. The studies demonstrate compelling data suggesting that poultry has a major prevalence of C. difficile within organisms' wastes compared to cattle, sheep, goat, camel, or buffalo. The study is suggesting that birds are representing a threat to public health, since their feces can easily be spread across a city or a community, especially in countries in which animals such as this kind interact with humans on an everyday basis. This potentially leads to the spread of the pathogen of interest, causing infection cases in large volumes. Here, in this data, showed in Table 1, the genetic information of poultry such as chicken, ducks, and quail were studied. The genes of interest were chosen due to its sensibility to C. difficile toxin typing. Results show that strains isolated from broiler meat contains genes that synthesize A and B toxins of C. difficile. The pathogen was extracted from various parts of chicken samples, demonstrating that in chicken meat, ribotypes are present, which are virulent factors that cause major intestinal problems, such as severe diarrhea. Besides C. difficile spreading across entire communities through feces, this pathogen is also using meat as another vector of transmission, which is a concern for human health, causing more cases of life-threatening diarrhea. In other studies, scholars claim that a potential therapy against C. difficile could be the transplantation of fecal microbiota from immunocompetent individuals into subjects that present infection of the pathogen. Studies reveal that the transplantation of such organisms can lead to the decrease of C. difficile from the guts, and that it is more promising than antibiotics. However, safety parameters need to be revised in order to rule out any possible risks. As already discussed, hospitalized people are prone to acquire C. difficile infection. Studies reveal that in hospitals, many patients start presenting diarrhea symptoms, in which, after revising testing samples, the specimen shows to be present in a range of 47.3% to 82% of the cases of diarrhea. This is compelling evidence that hospitals can act as vectors in which the disease can be easily transmitted. However, many factors can deviate the resulting cases of infection, such as the overall disinfection procedures of a hospital, and human habits, such as if employees are using the correct methods of hygiene. In other studies, they have revealed that depending on the country and the organism that acts as vectors for C. difficile, antibacterial resistance can be different. Data in Table 2 demonstrates that antibiotic susceptibility of C. difficile against ampicillin is low, meaning that the bacteria has acquired antibiotic resistance against the antibiotic. However, data also shows that C. difficile has high susceptibility to metronidazole and vancomycin. Scientists are still working on the effects that drugs such as fidaxomicin, meropenem, and piperacillin or tazobactam have over C. difficile. Interestingly, scholars claim that even though C. difficile is treated with vancomycin, the organism is already developing resistance to the drug. Nonetheless, this is not representing a major problem yet. This is only suggesting that a novel treatment will be needed for the future to fight against an infection of C. difficile. In addition, low resistance to tetracycline, clindamycin, and moxifloxacin was presented by the data. However, other studies have demonstrated that C. difficile can acquire high resistance against the later mentioned antibiotics. Novel epitope vaccine against C. difficile. The following article, Immunoinformatics Approach Towards the Introduction of a Novel Multi-Epitope Vaccine Against Clostridium difficile, by Tan and et al., introduces a novel vaccine that shows to be promising therapy against C. difficile. The vaccine was built under computational techniques. Further, the targeted proteins CDEC, which inhibits spore fertilization of C. difficile, and FLE-D, which inhibits the spread and settlement of the bacteria, were elected to design the vaccine to implement immune activity against spore germination and spread of the pathogen. The following epitopes were chosen in order to build the vaccine: 5 cytotoxic T cell lymphocyte, 5 helper T lymphocyte, and 7 B cell linear epitopes. In addition to increment immune response of the vaccine, an adjuvant was added. Further, platforms such as Prabi and Raptor X were used in the design of the 2 and 3-dimensional models. Additionally, molecular dynamics were also revised to study the activity of the vaccine against the bacteria. Finally, results show positive responses against the fight of C. difficile. However, more studies need to be carried on to fix details. Thank you for watching, and see you next time.